Hi there. In this two-part series, I'll talk about the evolution of ribosomes and the genetic code. As you might expect, it's a bit complex because it's been evolving for over 4 billion years. So I've had this as one video or one lecture, but it's become so large that I split it into two lectures, and this is part one. This is a summary slide of the evolution of molecules involved in translation, both ribosomal RNAs, the tRNAs, mRNAs, and ribosomal RNAs, as well as the aminoacyl tRNA synthetases up here, class 1 and class 2, which may have been originally RNA molecules also, but very early on that function was taken over by proteins, at first very small proteins, and they got larger and larger and became more diverse to charge, that is, add the amino acids to various tRNAs. So the first phase was here, greater than 3.8 billion years ago. And the tRNA consisted primarily of just the CCA tail, which was where the amino acid was added. The class 1 and class 2 aminoacyl tRNA synthetases may have been very small and possibly RNA molecules at the beginning. mRNA was a short piece of RNA. Small subunit was quite small and probably functioned to move other RNAs around already. And the large subunit consisted of the beginnings of the peptidyltransferase function, that is the enzymatic portion of the ribosome that adds the amino acids together or to the growing polypeptide chain. The small triangles or darts in these indicate points where pieces of RNA seem to have come together, possibly by being joined by introns or other enzymatic RNAs. In phase 2, about 3.6 to 3.8 billion years ago, the AARSs clearly were proteins by this time. The acceptor arm was fairly well developed, and the RNAs apparently were double-stranded. The small subunit and large subunit continued to grow so that the fidelity of translation increased. By phase 3, which now you clearly have DNA organisms, bacteria and such, and probably in phase 2 also simple bacteria, the aminoacyl tRNA synthetases are becoming larger to handle more complex tRNAs, and here's the tRNA has doubled in size, gaining the T pseudo-U C arm or T domain. The RNA, the mRNA, has become larger but still double-stranded, and I'll talk about why that probably was double-stranded. And then the small and large subunits begin to increase further, having an A and a P site for the tRNAs. In phase 4, the diversity and sizes of the class 1 and class 2 AARSs continue to increase. The tRNA doubles in size by this time, so you have fully developed the DNT domains as well as the anticodon domain. And also the mRNA has become single-stranded again, probably so that it fits easily into the ribosome and efficiently translates or efficiently is translated by the ribosome. And then the large and small subunits continue to grow, adding on piece by piece by piece. Also, introns certainly appear by this time, as indicated by these triangles. And by phases 5 and 6, everything has grown further and become more complex. More discrimination is gained by the ribosomes as far as when and where they translate what particular mRNA, what tRNAs they use, and the efficiency appears to be much greater. They also associate with a larger number of proteins, which aids in the discrimination of when and where to translate particular mRNAs. The tRNAs, either at this point in phase 6 or before, are able to exchange pieces or arms with other tRNAs, so that increases the diversity of those tRNAs as well. So by phase 6, you clearly have bacterial ribosomes and archaeal ribosomes, and not too long after that, by about 2.7 to 2.2 billion years ago, then you have eukaryotic ribosomes as well, which essentially the central part of the ribosome becomes fixed pretty much as far as its function and sequence, and then the outer fringes of the ribosome continue to evolve new little sections, again for discrimination of translation. So here you can see some of the differences in the complexity of a bacterial or archaeal ribosome versus a eukaryotic ribosome. So bacterial ribosome is a small subunit, has 21 proteins in the small subunit ribosomal RNA, which is about 1,500 nucleotides in size. And again, those proteins aren't doing a lot in the ribosome other than holding the RNA in its functional form. And then there's a 50S ribosome, which has about 34 proteins, again holding the large subunit 5S and 5S RNAs in their functional forms. And those are about 2,600 nucleotides and 120 nucleotides each. And that makes up the 70S ribosomes. Archaeal ribosomes are a bit larger than this. And they're on their way sort of to becoming these eukaryotic ribosomes, which have about 33 proteins and a small subunit ribosomal RNA of about 1,900 nucleotides, and the small ribosomal subunit, which is 40S, that is the Svedberg units. And then the large subunit has about 49 or 50 proteins and includes the large subunit rRNA, 5.8S rRNA, and 5S rRNA. And again, those proteins are holding those RNAs into their functional form. So in eukaryotes, the small subunit is about 40S, the large subunit is about 60S, and totally together it makes up an 80S ribosome. And again, the central parts of the large subunits and small subunits are very much the same, including quite a bit of sequence conservation, which is unusual for RNAs. But because of that sequence conservation, sequences from bacteria, archaea, and eukaryotes, the ribosomal RNAs, can be compared, so you can make large phylogenetic trees from these sequences and compare things that have been separated by billions of years. For instance, this fork here is about 3.5 billion years ago, possibly a little more than that. This branch separating out the archaea is about 3 to 3.5 billion years ago. And then going out to this branch that was an archaeal line, becoming the eukaryotes, that branch starts at about 2.5 to 2.4 billion years ago. So as I said in a previous video, the small and large subunits both have sites for the tRNA at the A site, at the P site, and the E site. And furthermore, the large subunit has a tunnel going through it where the growing polypeptide is pushed, and then it eventually comes out through that tunnel. That tunnel actually was there very early in evolution, and it acted to hold the growing polypeptide onto the large subunit. So it had a function even from the beginning. As the ribosome has gotten larger, the sequences around that had to continue to fold in a way that retained that tunnel. And so the tunnel's a lot longer now than it was 3.5 billion years ago. So here's a schematic of translation on the ribosome showing the A, P, and E sites, and then the tRNAs coming in, aided by hydrolysis of GTP. So you can think of the mRNA as moving this way or the direction of the ribosome moving that way. It's about the same. And the first codon then is the AUG, which has a methionine on the tRNA for that one. There's a shift by 3 nucleotides, that movement caused by the hydrolysis of GTP and the action of the small subunit. That shift also moves the tRNA in the A site over to the P site, and that leaves the A site open for a new tRNA with a different amino acid on it. And then the enzymatic peptidyl transferase function of the ribosomal RNA links those together, and by doing so, it detaches the amino acid on the tRNA at the P site, attaches it to that on the A site, and then things shift over. So now that tRNA is without an amino acid, moves to the E site, and is shuttled out of the ribosome. And then there's continual shifts then from the tRNA moving from the A site to the P site, transfer of the polypeptide from that tRNA onto the other tRNA. So you're constantly adding and shifting that polypeptide over to the P site, the tRNA in the P site. But it's initially adding to the tRNA from the A site. And that continues until a stop codon is reached, and there's no tRNA for that. So another molecule called one of the release factors fits into that site, clips the polypeptide off, and then starts a process with other release factors to pull the mRNA off, as well as pull apart the large and small subunits, and then they can start over again. So in addition to the peptidyl transferase site, the P and A and E sites had an evolutionary process. So initially there was just a P site, which also contained the peptidyl transferase activity, and that tunnel, at least the starting of a tunnel, probably just a circle at that point, that could hold on to the growing polypeptide while the peptidyl transferase function found a tRNA that contained the corresponding amino acid and added those together, and then that emptied the P site for movement into the P site by that tRNA that now held a longer polypeptide. Probably more than 3.8 billion years ago, there was a duplication of the tRNA binding site. So you can tell that because the current sequences around those areas, the P and the A site, look very, very much the same, and they're about the same size of about 110 nucleotides. So now this increased the success or efficiency of that process, because then the tRNAs at the A and the P site were held in place so that the peptidyl transferase could link them together, the amino acids in those positions together. The E site is somewhat different, so it doesn't look like it was a duplication of the P or A site, but it's fairly close. So it may be a more divergent version of a tRNA binding site. After that, then layer upon layer of other regions of the ribosome were built around that, and I'm just talking about the large subunit in this case. And so you've had more and more discrimination, more and more other proteins associating with the ribosomes to increase the diversity of the translation system. So a few years ago, a model for ribosome evolution was built based on sequence comparisons. So very early on, back here at A, you have just that protoribosome that has the peptidyltransferase function, but little else. It has the hole that will become the tunnel. And then regions were built up around that called the foundation, and little by little those got larger, and that included the A and the P sites, and eventually the E site. And then you have this ribosome becoming larger and larger. In this diagram down here, you have the large and the small subunit of the ribosome as well. This has been called the onion model because apparently the ribosome has pretty much been built from the inside out. So very early in evolution, you just have the core region here. And then little by little, layers of RNA were added on to that ribosome to increase fidelity, to increase discrimination of translation and other functions. The process of translation using a genetic code is another part of this entire system. And if you just look at the genetic code, this is a table of the genetic codes, starting with the first position here of the codon, second position of the codon, and then third position of the codon. So phenylalanine up here would be encoded by UUU, which on the anticodon of the tRNA would be AAA or something similar to that. But you can already see the structure here of these orange colored boxes for those amino acids. Those are nonpolar amino acids. And then the purple ones are polar amino acids. The blue ones are basic, having a positive charge. And then the ones in green are acidic, aspartate and glutamate down here. So there's a structure here that says something about the evolution of the genetic code and the process of translation of mRNA into protein. So this is the same genetic code, but a few details have been added. So if you look down here in the bottom row, you can see that these R groups on these amino acids are very basic and simple. Here's valine with 3 carbons, alanine with only 1 carbon, glycine just has a hydrogen, no carbons, and then aspartate and glutamate have 2 and 3 carbons between them. And then if you go up on this side, you can see that the structures get generally larger and larger. And if you go into this upper right-hand corner, the structures get even larger. There are more ring structures and things are more complex. So you pretty much go from simple down here to more complex up here, and the most complex are up here in the upper right corner. So here's a chart of those amino acids going from the simplest, which is alanine, valine, isoleucine, leucine, and then getting more complex ring structures over here with phenylalanine, tyrosine, and tryptophan. The amino acids here are polar, so they have usually an OH group, an amino group, or a carboxyl oxygen, which gives them a partial charge. You have the alpha helix breakers here, essentially, such as cysteine, selenocysteine, glycine, which is pretty much a placeholder, and proline, which is actually an amino acid, spelled with an I, immuno rather than amino, and it introduces kinks into the polypeptide chain. So these ones are pretty much structural in one way or another. These ones down here produce mainly alpha helices and beta pleated sheets, as do these polar ones here. And then the ones up here are charged. So arginine, histidine, and lysine all have a net positive charge on them. And the negatively charged amino acids are aspartic acid and glutamic acid, or aspartate and glutamate. But remember, 10 of these were available on early Earth, so that includes alanine, valine, aspartate, glutamate, glycine. And incidentally, those ones are on the bottom row of the genetic code, which appears to be the beginning portion of the genetic code. And then proline, threonine, serine, leucine, and isoleucine were also among those original 10. And to show you where those 10 are, they're here. The red ones are those ones on the bottom row, so you already had these ones down here that could form alpha helices and beta pleated sheets. You have glycine, which is pretty much a placeholder and freely rotatable, and then a couple of groups that could move electrons and protons around. The other ones that were available at that time were these ones. Again, now we have nonpolar and polar groups available, so things that would fit into a membrane and things that would float in aqueous environments. And also proline, which would introduce kinks into proteins, polypeptides. And so you have pretty much examples, at least, of all the functional types of amino acids available even from the very beginning. And so those were probably some of the first amino acids in ancient proteins, and I've already shown you previously that that's been confirmed in several studies now. So those smallest and simplest of the amino acids are down here, and those were clearly available early on Earth. So that implies that the first genetic code might have been a one-letter code consisting of a U standing for valine, a C standing for alanine, an A standing for either aspartate or glutamate, or both, and a G which would encode for glycine. So there could have been, instead of a triplet code, a one-letter code very early on in translation. So this is the one-letter code indicated over here, where one nucleotide stood for one amino acid. So the tRNA would stick to one of the nucleotides, and it would carry one particular amino acid, and then you could start building up using the peptidyl-transferase of the ribosome to build up a peptide, a very simple peptide. There could have been a duplex code, that is later, where now the tRNA is sticking to two nucleotides and bringing the amino acid in. But there's some evidence that the alternative was to build immediately a triplet codon from an RNA that could be read in both directions. That is a double-stranded RNA where if there was another strand here also, it could be separated, and then the RNAs could be read in one direction and the other RNA from that duplex read in the opposite direction. And that implies a triplet code already, where the third nucleotide is not so important, and that's what we see today. And I'll explain that before we're through. So this diagram here at the bottom, and I've shown this before in a video, where these are the frequencies of amino acids in the most ancient proteins. So the most frequent are these ones: alanine, valine, glycine, glutamate, and down here is aspartate also. So you have those 4 or 5 that are on the bottom row of the genetic code. And if you look at them, they could be encoded by A, G, U, and C here. The next nucleotide added appears to be a G in the duplex or triplet codon. So you'd have GA, GG, anything, GU, anything, GC, anything, and those would encode those. Later on, these shift a little bit, and so then G isn't used quite as much. But in the ancient proteins, you can see there's definitely a preference for that G as being the initial codon in either a duplex codon system or a triplet codon system. So the next step could have been something like this, where now you have 2 nucleotides or 3 nucleotides. Either one will work, especially if that third nucleotide in the codon doesn't really matter. It can be any H, E, C, or U. But anyway, you can have GU standing for valine, GC for alanine, GA standing for aspartate or glutamate, and GG standing for glycine. Now, whether there's a codon consisting of 2 nucleotides or 3 nucleotides, if the third nucleotide in the triplet doesn't matter much, then the number of amino acids can grow from the 4 in a 1 nucleotide codon system to 16 in the next system, having at least 2 nucleotides in the codon. And this starts to resemble the modern table for the genetic code. And it may have been this type of system as long ago as 3.5 to 3.8 or maybe even 4 billion years ago. Because of the availability of the amino acids at that time, then this part of the table starts growing. So you have isoleucine and threonine. Note that methionine isn't there yet. It was actually a late arrival. And notice that it wasn't the ones that were available very early on Earth. So it was added much later. So adding on to that previous graph, so up here above, that's that one indicating the most ancient protein components, that is, the frequency of amino acids in those most ancient proteins. And those are proteins that are in every organism, so we know they've been around a very, very long time. And so here are those 4: alanine, valine, glycine, glutamate, and aspartate. If you look at more modern proteins, then you see that the frequency of G being the first nucleotide in the codon decreases. And the things in red here are amino acids that are decreasing in frequency, not necessarily in number, but in frequency. And the green ones are those amino acids that are growing in frequency, and these are some of the more complex amino acids. So the genetic code is getting more complex, and many of those amino acids can only be formed biologically and were not available very early on Earth. So then if we look at the evolution of the genetic code, we start with this bottom row here with valine, alanine, etc., and then move up in this direction with this large arrow, because those are all the ones that were available early on Earth. Then there was some movement in the genetic code, both upwards from aspartate and glutamate to glutamine and asparagine, as well as movement over in this direction into some of these polar amino acids. And then finally, the last steps were along this top row, and especially this upper right-hand corner, where you have tyrosine, cysteine, and tryptophan, and histidine as well, which are some of the more complex amino acids that can only be produced in biological systems. So looking in a little greater detail, including the start and stop codons, the original start codon probably was valine, and in some mRNAs, even today in some organisms, a valine tRNA is used as the start codon. And then it looks as though the beginning amino acid was either alanine. Some people have said glycine was probably the first one. The evidence I've seen points to alanine, and then things spread out from there. Then the genetic code expanded to include isoleucine, including an isoleucine that was a start codon, threonine, and then 2 stop codons. Before that, there may not have been a stop codon, or all of these could have been stop codons, or translation could have gone on just till the end of the short mRNA. So the code continued to move upward. Proline was available at that time, so now you could have hard kinks in the polypeptide chain. Arginine seems to have appeared about that time, although that wasn't a natural amino acid that could only be produced in organisms. And then there appear to have been quite a few stop codons by that time as well, some of which are still used today in different systems. But ultimately, a triplet codon was developed, and the P site expanded to a P and an A site, so that then there were 2 tRNAs next to one another, increasing the efficiency of joining those 2 together, the amino acids together, by their peptide bond. So next, it would proceed from here to here, where more of the amino acids are added, including leucine, serine, and notice leucine had expanded into several places, and all of the start codons were over here that were used, and some of those are still used in different systems, including down here ultimately the AUG that eventually encoded for methionine, and then there were quite a number of stop codons as well. And next, there was a little more filling in of different areas, including gaining lysine, histidine, and reducing the number of stop codons and the addition of methionine. These numbers in parentheses indicate the frequency, the relative frequency of each one of those amino acids. So down here you have number 1, highest frequency, number 2, number 3, number 4, those are those ones on the bottom row. Adding isoleucine is number 5, and then going over to arginine 6, etc. So note that methionine is number 15. It was added very late in this translation system. When I first learned about this and read about it, I was a little bit surprised that methionine was such a late arrival. Because it's almost a universal start codon for most organisms, most mRNAs in organisms, although it's not completely universal. There are other start codons that are still used by bacteria, protists, and other organisms. And the final additions are up here: tyrosine at number 18, tryptophan at number 19, and cysteine at 20, and there are some tryptophan codons that are encoded by this stop codon instead. And the stop codons up here have kind of been squeezed into one corner, and in some systems they are actually used as codons for particular amino acids, including selenocysteine in some systems, which is essentially the 21st amino acid that looks like cysteine, but instead of having a sulfur, it has a selenium atom in that position. So here's the genetic code, and this is one model based on actually a lot of data as far as frequency of amino acids, size of the amino acids, and what sorts of evolution you would expect based on shuttles off of the TCA cycle, off of other biochemical pathways, because a lot of these amino acids now are either used in the TCA cycle peripherally, or they can be formed from the TCA cycle and other biochemical pathways as well. So you can make up a fairly good story that this model for evolution of the genetic code is probably pretty close to what actually happened. Okay, so that's the end of part 1. As I said, it becomes very complex in a hurry. So there's a part 2 to this where I present a little more information, actually a lot more information, about how some of these systems evolved. So I'll see you in part 2. Thanks very much for listening and watching. Talk to you later.